Next in the program, we're thrilled to have gathered a panel here of leaders in biotechnology and biomedical research who are advancing the cutting-edge interventions that are going to be extending the healthy years of life in the near future by targeting fundamental biological aging processes. And we're also honored that Jamie Metzl, an Atlantic Council Senior Fellow for Biotechnology and National Security, has joined us to moderate this discussion. As a novelist, blogger, syndicated columnist, media commentator, and expert in Asian affairs and biotechnology policy, now that's a pretty broad field. I must have to say that's impressive, Jamie. He's testified before Congress on the national security implication of biotechnology in the genomics revolution. His novel, Genesis Code, deals with the issue of human genetic enhancement in the context of the future U.S.-China rivalry. And his new book, which is on your table there, addresses issues of aging that seem more and more like they're not science fiction, but they're science fact. So please welcome me and join me in welcoming Jamie to the book. Pardon? Ah, okay, so look under your coffee cup to see if you're the big winner. Mark, don't change my coffee cup. Oh, anyway, please welcome Jamie Metzl to the stage.
Thank you very much, Steve and Stephanie, and thank you for all of you. So now we have a little bit of chaos of everybody looking under their coffee cups. My friend Nir, who came early, said that he was sneaking around looking under coffee cups and realized that I had won. And so he selected somebody. We can't figure out who it was at our table. But thank you all very, very much. It's really just a pleasure and an honor to be here with all of you in such a beautiful place and in such an incredible moment, not just for this field, but for scientific research and medical research, and really for all of humanity. Because we are at this inflection, this turning point, where really after 4 billion years of evolving by one set of rules of random mutation and natural selection, we are turning an evolutionary corner with all of these new tools that we've been talking about and will talking about that are unlocking in many ways the code of life. It's a truism that when Watson and Crick identified the double helix structure of DNA, they identified there was a manual of life. And now with sequencing of DNA, we're identifying how to read that manual. And with gene editing, we're learning, at least in preliminary stages, how to write the code of life. And when we think about technologies that are writable, readable, and hackable, obviously today we think about information technology. But what we're seeing in many ways is this transition, which will take decades and centuries of seeing biology as a new form of information technology. And that's, as with all of the work that AFAR has supported, it's unlocking in many ways this code of life and unlocking this tremendous human potential to do all sorts of things once we have that knowledge. And we can talk about this in scientific terms, and that's what many people here do, and that's the critical work that's being done. But we need to take a step back and say, well, what does this mean? What is the work that Ned was talking about, and many of you are working? What does it mean? What does it mean if we extend health span by 1 year or 2 years or 3 years? How much poetry will be written that wouldn't be written? How much time will people spend with grandkids? How many inventions? How many, all of these magical things that human beings do, how much more of it will we be able to do? So I'm a science fiction writer, as you know, and so when I think about these issues, I like to think about this moment in the context of stories, which is why I write these books. But imagine that we had a time machine and traveled 1,000 years into the past, and we kidnapped, which is a mean thing to do, but it's science fiction, a baby, brought them back to today and placed them with a family. And of course, that kid would grow up to be an adult, just like indistinguishable from anybody else. but go 1,000 years into the future, bring that child back, and that child would be, in many ways, by our perspective, a Superman or Superwoman. They would have capabilities that are very different from ours. They would live healthier, more robust lives. They would have all kinds of genetic capabilities that right now are maybe outlying traits of humanity, or animal traits, or traits that don't yet exist in humans or animals, but they can be built with the basic building blocks of life that we have. But one of the changes that those people will have will be that they will live longer and healthier and more robust lives than we do. And in many ways, that's not a revolutionary thought. We as our species, for a very long time, have been imagining what immortality might look like. We all know in our Western traditions of Gilgamesh and Methuselah. I just came from China yesterday. In China, there are these mythologies of magic mushrooms. My girlfriend Malika is here from India, and India has the mythology of soma, which you would ingest and help you live longer. And what is happening, though, is that over time, all of those dreams are becoming real. And they're becoming real more and more every year. As many of you know, at the time of the Roman Empire, 2,000 years ago, average life expectancy was 25. In the Middle Ages, in Europe, it was about 30. At the turn of the beginning of the 20th century, it was late 40s. And now in the United States, it's approaching 80, and it's longer in other places. And so this obviously is a big change, but it's not just a change in lifespan or even health span or average lifespan or health span. It's also a change in people's ideas. And so that if somebody died when they were 50 2,000 years ago, nobody would say you were robbed. I mean, everybody would always like to live longer. But now if somebody dies when they're 50, we say that they were robbed. Right now, if somebody gets cancer when they're 80 or when they're 90, we think it's terrible, but we think it's within the normal flow of things. But there's no reason why that would need to be the case. And that's the work that we're all here together in order to achieve. And our science of longevity has been advancing. A lot of the gains have been because of not research directly into longevity, but of work done on basic sanitation and healthcare and nutrition and all these other things. But really, over the last century, perhaps, we've begun to make, as you all know, some very significant progress. We were talking at our lunch table about a major innovation that happened about 1890, and that is the injection of ground monkey testicles, which in 1889 people thought that was the hottest thing. As a matter of fact, Nir Barzilai is doing a double-blind study of whether ground monkey testicle injections work. So if you'd like to be part of that study, Nir is at table 8, and you can sign up with him. What's that? Yeah, and then in 1920, as some of you know, there was this major innovation. They said that no, ground monkey testicles is not the way to do it. You have to sew in a full goat testicle, and that unfortunately didn't work. But if you'd like to be part of that study, again, Nir has made his good offices available. But now we're on the verge of just these incredible breakthroughs. And I thought Ned's speech was terrific, that we have all of these little pieces. And many of you represent these little approaches because aging is so complex and systemic. We're all, in some ways, the blind people trying to touch different parts of the elephant. And that's really important. And the question is, how do we bring all of these pieces together? How do we bring these pieces together in a way where we can tell a story and translate that into a course of action that extends healthy human lifespan on a systemic way and unlocks the type of poetry and potential that I spoke about a moment ago? And so we kind of take a step back and say, well, how can we think about this problem in big, broad terms? we have to say, well, what are the clues? What are the models that we need to look at? And the ones that we do, of course, are human and animal models. And from a human model, when we think about how we evolved into this form over 4 billion years, it gives some directional ideas of different places where we might look. The good news for longevity work, as I see it, is that evolution hasn't really selected for or against longevity as we are thinking about it, because getting to these ripe old ages, at least on a broad population level, was something that was relatively rare, not impossible, but relatively rare in the past. And so evolution didn't select for it or against it. If we as a species had a problem that too many of our children were being eaten by saber-toothed tigers, humans would have evolved exoskeletons like lobsters. And if we had a problem that too many parents were being eaten by saber-toothed tigers, we would have evolved a process where our children could be born like Komodo dragons, and you're on your own from day one. If you're a Komodo dragon and you run into your mother 5 minutes after you're born, you're going to get eaten. But we haven't evolved. We've evolved in the ways that we have. But grandparents who are 90 years old or 100 years old, that was never part of our evolutionary selection process, because most grandparents never got to that age. So if the grandparents got carried away by a saber-toothed tiger, it was bad for the grandparents, but it didn't really affect our evolution as a species. Another thing we've talked about, calorie restriction, is that in our history, it's 60,000 years ago, again 300,000 years ago, our species was right on the verge of complete annihilation from starvation. And the people who survived, these small numbers, in 60,000 years ago, we were down to under 10,000 Homo sapiens on the southern tip of Africa. The people who survived, it's the equivalent to the fruit flies that survived in the studies where there's not enough food for everybody, and the fruit flies, 90% of them die and 10% survive, and then you do it again and again and again. The humans that survived were the ones who had the capacity to live on the equivalent of screensaver mode. And that's why we all have, when we talk about why does calorie restriction work, in my mind, there's something deep in our genetics that we were the ones, our ancestors were the ones who were selected for this ability to live in these environments of tremendous scarcity. Other places we can look in human models are looking at the centenarians, like Neer and others are studying, to say, are there genetic variations between the people who are living an extremely long time, and what do those mean? Or groups of people, like Dan Buettner and others have studied in the blue zones. What is it about groups that helps them live longer? Probably not a biological selection, but lifestyle selections. And then in the animal models, there's a lot that many people in this room are learning to say, well, what are the differences between animals within the same species or in related species, like different types of clams or the naked mole rats that many people are studying? What's the difference between related animals, that some live longer and some live shorter? What is it about these other types of animals, like the jellyfish, that have this seeming ability to live forever? Those of you who won my book, and you'll see there's a lot about the immortal jellyfish in this book, but what are these clues from the animal kingdom that we can learn from, that can give us clues into how we can live longer and we can live healthier lives? But this, as I said before, is all just a tremendous, complex puzzle. And the exciting thing of living at this moment, when our species is networked to each other like we've never been before, is we have ability to supercharge this process moving forward. And we all talk about exponential change. And with exponential change, you're all familiar with Moore's law. But as our biology becomes a form of information technology, then our biology itself is on a Moore's law curve. And that's why, coming back to what Ned and others were saying, that's why the work of AFAR, and I'm always confused, and I don't know if there's a complete answer, whether it's AFAR or AFAR. Either one. Why AFAR, I'm going to say, because it sounds like it has, that work is so important, that we are on the verge of this exponential change. And so just a little push in a direction, it's like doubling your pennies, just a little push in the right direction or in a different direction has the potential to lead towards just fundamental and significant and remarkably transformative change that will unlock all of these kinds of wonderful aspects of being human. So with that as an introduction, I would like to call up the panelists, and while they are coming up, I will give a short introduction. So panelists, I've got one panelist. Yep, good. Great, and you have terrific. And you have everybody's longer bios in your booklets. Yep. So Nir Barzilai right here is principal investigator of the Targeting Aging with Metformin TAME trial. And we're going to speak about that in a moment. And he's the AFAR Deputy Scientific Director and Director of the Institute for Aging Research at Albert Einstein. Judy Campisi, who was mentioned before, is scientific co-founder of Unity Biotechnology and former member of the Scientific Advisory Board of Sangamo and a professor at the Buck. Ned David was already introduced. Jan van Dersen, co-founder, again, of Unity Biotech and director of the senescence program in the Robert and Arlene Kogod Center of Aging at the Mayo Clinic. And Eric Verdin has served on the scientific advisory boards of Elixir, Certris, and Nokia and other companies, and president and CEO of the Buck. So we will start. Hold on. We'll start just for setting our baseline, and just with a few words. We're talking a lot about aging, and yet aging is in many ways hard to define in a specific way. Maybe just a few words, and we'll start with you, Nir, and just go across. What is aging? How do we define aging? And how can we measure aging?
In few words, it's the couple who lives in Midwest United States in a nice house, and the woman comes to the husband and says, "Honey, why don't we go up and make love?" And the husband looks at her and says, "Sweetie, I cannot do both." I think that's the functional definition of aging.
Yeah.
Let's move on.
And we can do a comic round, and then we can talk about biomarkers and definitions on a second round. Why don't we actually do that?
So I'm not funny that way. So I've heard Nir's anecdote of that one before, and Judy has some really good ones. I'm not going to steal hers.
Yeah.
I'll give you my serious answer. To me, I tend to think of it when I think about aging. I contrast two things. I contrast the seemingly eternal phenotype of the germline. That this portion of each of our biologies that seems to move across generations in this unaltered way. It's a piece of all of us that is immortal. And when I think about what is aging, it is all of the molecular contrasts that we exhibit as over time that differ with that. And yeah.
Good. I think if we knew what aging was, I think none of us would be here, probably be hiking in the Golden Gate Park. I am humbled by the complexity of what aging is, and I think we are all at this point touching different parts of an elephant. You know, some are touching the legs, some are touching the trunk. I don't think we have a really good understanding of what aging is, but we're getting hints. And one of recent finding that I find quite interesting is this whole idea of this epigenetic clock that tells us of some underlying principle that allows you to predict, how old you are. I think we're we're nowhere in terms of understanding what it is, and I think I'll leave it at that. And more reason to continue working on this.
I'm with Nir. It's like pornography. You know it when you see it, right? And you feel it when you know you're old. I mean, at least I've been told. I have no idea. Yeah.
Well, I have a more scientific approach to this, maybe. So I'm a molecular biologist, and the way I see aging is I guess a gradual change in molecular and cellular processes over time, and at each period maybe trying to find a new balance, homeostasis, and at some point that process stops, and then we die.
Yeah.
So can I?
Yeah.
What a buzzkill.
Wow.
Yeah.
Yeah, Judy, please.
Yeah, so I think it's also important to dissociate aging from death. And,
Yeah.
and so aging is this process that we all hate, and death is inevitable. I mean, we're all going to die, even if we had so-called immortal germline cells throughout our entire body. At some point, we're all going to die. So I think it's really important to distinguish between those two. And what we're after is to understand aging, not necessarily death.
Absolutely. And aging, and I think that everyone in this room understands this difference between health span and lifespan. And that's one of the great challenges that we have. We know that lifespan is creeping up. But if lifespan goes faster than health span, then you get a lot of people in old age homes and living these kinds of lives that nobody aspires to. But when we talk about all these, you're right that in many ways, and we saw the mice in Ned's presentation. And so it's intuitive. You kind of see these mice moving around, and you can kind of look at the bones, and you can look at all sorts of things. Do we need to have a standardized definition of the biomarkers of aging in order to benchmark all of these different pieces of the puzzle? Like what's working, where are the biggest gains to be had? And if so, how do we get from here to there?
I think this is one of the key questions of the field, because we're entering a stage where we are all wanting to translate some of the findings that we've made into patient clinical trials. And I think you have both Nir and Ned, who are both confronted with this. How do we measure the rate at which we age? How do we measure the efficacy of our intervention? I think, and they've taken two different approaches. I won't speak for them, but I think it's going to the identification of biomarkers that allow us to quantitatively assess the rate at which either a person or individual organs are aging is really one of the key areas that we are focusing on, and the field will need to focus on.
So maybe Nir and then Ned, why don't you talk a little bit about your approaches to this?
So AFAR has been involved in I don't know how many conferences on biomarkers for aging. We all try to do it. We try to do it in many ways. There's always somebody at the table, a geriatrician, who said, "What do you mean biomarker? You just ask the subject to get out of a chair and you'll see if they're age or not." And we never could really do that. By the way, the FDA wants to have biomarkers. The FDA wants us to have a test when we're 50 and to see if we're biologically 40 or 60. If we're biologically 40, we don't need colonoscopy, right? So this is really important, but it became even more important because now we're testing treatments. And we want the biomarkers not only to reflect our biological age, but to reflect if the drug is actually doing about turning back our biological marker. So this is the challenge. I just want, some of you have been in my lecture today, and I think this is how I've changed my view in the last year. Senolytics, rapamycin, metformin, you give them to animals and you see they have a lot of effects and a lot of useful effects. And it's kind of clear to me now, and we did this study on metformin where we took biopsies before and after in people, the true aging targeting drugs are going to affect a lot of biomarkers. And that's maybe why we were so confused, because we never agreed on biomarkers because there are a lot of biomarkers, and maybe it's the information technology that will help us show a cluster that gets better or not?
So I'll talk about aging biomarkers essentially in two buckets. I'll talk about what we do with senolysis. That's when we remove senescent cells. So, and I think that's the much easier to explain story because it's something we're doing today. So if you imagine you have a disease that's caused by the accumulation of senescent cells, you'd like to be able to identify patients that have them. So we do this, and in fact, we just completed what we call a phase 0 clinical study where we enrolled 30 patients and looked in their knees for their senescent cell burden and measured it very literally using very well-established markers. And the idea is that you would then be able to, as you test patients, see who responds to your drug and who doesn't, and see if there's some sort of correlation to their senescence burden. And if you learn that's the case, in the future, you would imagine you would go to the doctor and they would take either a drop of peripheral blood or even a sample from your knee, and they would say, you know, you're a good candidate for the drug, or no, you're not. So we do that for every one of our indications to go after, try to come up with a way to assess senescence burden. So that's pretty easy to understand. Now I'll go to the second bucket, which is the harder to understand stuff. Remember that big circle I showed with the question mark in it? Where the fundamental, hopefully the ticking clock that is aging resides? Finding markers that reflect that, I believe, will be among the most powerful tools to do fundamental biological discovery. Put aside clinical trials, right, in people. Just having a tool that could tell you how old a creature is, where is it on its aging trajectory, or even cooler, what is its rate of aging at that moment? That's kind of a heavy one, okay? If you had such markers, these things could be leveraged across so many different biologies, and I think will become fundamental to the next few decades of insight. An example is this ticking methylome clock, but there'll be other ones like that. For you guys that don't know what the methylome clock is, it's super cool. It's a technique where we can go to anyone in this room, look at 353 spots on your DNA, look for the methyl group, and we can tell within 1 year how old you are. It's pretty neat.
So
That's assuming you don't have a birth certificate.
there's always the old-fashioned way. So you guys down here, what's it going to take to get there? I mean, what are the impediments, the scientific impediments from where we are to getting that kind of understanding of the biomarkers of aging, and how can we overcome those?
So I think the biggest impediment is going to be the incredible diversity among people, creatures who refuse to inbreed. And so this is...
Speak for yourself.
Yeah.
Sorry.
Yeah. Actually, Ashkenazi Jews.
But I think every physician, anyone who's dealt with patients, knows that you know, you can have certain criteria by which you define a disease or you define even aging, but it's going to vary enormously from person to person. So we're going to need big data, big bioinformatics to help sort through the genetic diversity as well as the biological diversity of what we call aging.
And that's why this moment is so exciting, because it's not just that this is a revolutionary moment in the research in the aging field. It's so many different fields, big data analytics and machine learning and artificial intelligence and all of the high throughput machinery that speeds up all of these processes. Eric, you're going to say something.
I just wanted to amplify a little bit on what Judy was saying. Quite often, a lot of hype has been generated for what we've been able to do in simple animal model system, where we can double lifespan with small molecules and mutations. Being a physician and realizing how heterogeneous the human population is, it just adds a whole other level of complexity to what we're proposing to do. And quite often, people, if you look at someone who has a cholesterol of 300, any aging research is probably highly irrelevant, because they're going to be dying of a heart attack when they're 45. And I think this is the type of, there will need at some point an integration of both the basic research of aging and the physician who actually are in the trenches dealing with a whole series of other issues, high blood pressure and so on. And I think we will really deliver the promise of this field when we are able to integrate both the clinical research and the basic research. And I think this is really one thing that excites us.
Yeah, and that's why, I mean, to quote Near, that the mice keep betraying us. They keep living 5 times longer, and then nothing works for us because of this incredible complexity and diversity. So maybe let's talk about a few of the different approaches that you and others are working on. So we've talked a bit about senescence, but let's talk a little bit about that more. Maybe starting with you, Judy. Ned, in his remarks, talked about that cellular senescence. On one hand, it's a problem. On the other hand, it's a benefit. How do we think about the balance between maintaining the benefit and addressing the challenge of cellular senescence? And how do you see that translating both on a scientific level and a translational level as you continue on what Ned had begun in his remarks?
So it's even worse than Ned said. So we know that part of the reason why senescent cells drive pathology is because they're secreting lots of stuff. We also know that some of those things they secrete are bad. They cause what we call chronic inflammation, drives multiple age-related diseases. And some of the things that they secrete are good. They drive tissue repair and regeneration. So I think Jan and I are going to be in business a very long time because we still don't have a very good definition of what a good senescent cell is, what a bad senescent cell is, what the good secretions are, what the bad secretions are. There's just a tremendous amount of basic biology that we need to understand in order to be intelligent about applying some of the new approaches, which is to get the cells to go away.
So what does the scientific path look like from here to there? How do you get there?
So it's the way scientists do lots of things. We start marching in a direction, and then we keep our eyes open on the periphery, and we change direction when the results tell us we should be looking somewhere else. So we start by drilling down and understanding the biochemistry, the molecular biology, the cell biology of different cells, how they become senescent, why they become senescent, and what the consequences are. And that's 20 years of work just for me, and then probably 40 years for Jan, because he's younger.
It includes revising grants, what you said at the time?
That's a tough one here. No, but it's true. It's really basic. There's still a lot of basic biology we don't understand. And that's just senescence. As Ned said, there's the big question in the middle and all the other little things. So
No, we're going to get to the big. We have a long way to go. Yeah, we're going to get to the big. We're going to do some of the peripheries and then get to the middle. But Jan?
All I can say is that, if you remove senescent cells from a mouse at midlife, and you do that for the rest of the life of that animal, there's no detectable negative consequences. So there might be positive roles, but they could be minor.
But they're living in a cage, Jan. 5 to a cage. They're genetically identical. That's not
Sure. But
people.
we kind of also are, domesticated in some way, right? We live also in...
Speak for yourself.
This is a hot-smoking folk singer you're sitting next to.
And she's from California, and he's from Minnesota, right?
But I would say that we're also kind of living on support, right? We have healthcare, we have clean water, we live like a mouse almost, right? In a cage. And yeah, I would say so.
Yeah, exactly.
But so you know that. But whether removing senescent cells would have detrimental effects if you start when you're like 3 months old for the rest of your life, or whether it has any beneficial effects if you start when you're 80, we simply don't know at this point. And so that's interesting. To come back to your earlier question about aging or biomarkers of aging, I think what I find is interesting, where there's lots of opportunities, that if you are a biologist, you try to understand the biology of a cell, or maybe of an organ, of an organism, try to figure out how it works at the molecular, cellular, and organismal level. And we haven't figured that out. And how many scientists are there in the world studying biology or medicine. So what I think it takes to understand aging is to replicate all that effort, but now do it in old cells and old animals. Because from what we know right now, is that, you know, the DNA repair machinery works very well when you're young, but I'm sure it doesn't work as well when you're old, because of methylation changes in your genome, the components that are essential to drive that machinery are going to not be expressed at the same levels. So the machinery is probably not in tune anymore. So I think all these biological processes, they will get out of tune, and we need to just understand how that happens and how all these processes out of tune then still make a functional cell.
Yeah.
Right? I think that's the task that we as fundamental biologists need to do. And then all these biomarkers that we're talking about will just roll out like
if
no
we can
problem.
get to the unified theory of aging. So let's move on. Again, we're moving around the periphery here, but Eric, you've done a lot of work on mitochondrial rejuvenation. Can you talk a little bit about that as a strategy for enhancing health span?
So mitochondria are think about them as the power plants in the cell. So they are the places where we take nutrients and transform them in the currency that the cell utilizes as a form of energy. So they are critical in terms of maintaining cellular function at every single level. And there's growing evidence that as we age, our mitochondria become actually less and less effective at generating energy. And we know also in individuals who have selective mutations or problems in terms of mitochondrial function show accelerated aging. So this is an area, and actually we've published with Judy that one of the consequences of a dysfunctional mitochondria is increased senescence. So there's a link directly between mitochondria and the senescence process. So there are a number of approaches that are being taken today to try to alleviate this. One is probably one of the oldest theories about aging, the oxidative stress theory of aging that many of you are familiar with because you can walk into a Whole Food market and there are lanes of antioxidants. So this was a theory that was proposed about 50 years ago or so, and initially generated a lot of excitement, which was met by some failures in clinical trials. And I think one of the reasons for this failure is that people decided to use very broad-sided antioxidants, which actually block some of the good effects of the oxidative process, burning oxygen. So there's actually a revival of interest in that whole area, in particular from Martin Brand at the back has actually identified novel molecules that block oxidative stress, but in a very selective manner. And so these small molecules actually have incredibly interesting effects, which I think will bring this theory all the way back to the forefront. So that's one aspect. So antioxidants, the way you buy them at the Whole Food market, do not work. At least that's the consensus. But there will be new interest in doing this in a much more selective manner. And the preliminary results seem to be supporting that.
But it's good for the economy, to support the economy.
Yes, so yeah, exactly, for Whole Food market, and now which is Amazon. Second area is the role of NAD. So this is one of those currencies in the mitochondria that is really critical for intermediary metabolism. There's evidence that this NAD decreases during aging, and there are no efforts to show that supplementing in different forms NAD to bring it back to a more youthful level has actually quite protective effect in a number of model systems. So that's one area. The third area is the whole idea of trying to maintain the number and the function of these mitochondria by blocking or activating processes called mitophagy or mitochondrial biogenesis. This is, if you exercise, for example, your body responds by making more mitochondria, and this is one of the processes by which we think exercise actually exerts positive effects. So now there's the idea of generating small molecules or drugs that could actually elicit the same response, so trigger mitochondrial biogenesis, make more mitochondria. And let me see, I'm trying to think about the last area of work is nutrition. And what types of nutrition are more beneficial. It turns out if you're eating carbohydrates, are metabolized mostly independently of the mitochondria. If you're eating fat, actually it is metabolized directly via the mitochondria. And we know that the nutrient that one uses in different combination can have a profound effect on aging. That is, being a high carbohydrate triggers insulin and is associated with one of the major aging pathways. And so nutrients that go directly to the mitochondria seem to be more beneficial, so the burning fat, for example. And in this respect, one of the things that we study in my lab is the role of ketone bodies. Some of you have heard about a ketogenic diet. Ketone body beta-hydroxybutyrate is actually metabolized directly by the mitochondria. And we just completed a study that shows that an increased lifespan on animals that are on a ketogenic diet. And two other groups have found the same thing. So I think these are the 3 or 4 areas in which mitochondrial function, we're trying to go at it. And
I think it's a very interesting point that all of this is very important, highly scientific research, and some of it will lead to small molecule treatments, but some of it may lead to suggested lifestyle changes, some of which we already know that everybody should exercise and eat healthy food. But I was on a panel last week with Mark Hyman from Cleveland Clinic, and his whole thing is food is information that you are supplying to your body. And it may be that the interventions that we have are things to eat, different times to do different things. It doesn't necessarily need to be that high-tech research leads to high-tech interventions. It could be all kinds of low-tech things.
I think it's a critical point, because I think we're all thinking about drugs and so on, but there's a lot that we know already. And Jack LaLanne, some of you around the room know him, said at some point, nutrition is queen and exercise is king. Put them together and you have a kingdom. And I think this should be the foundation of everything we do, because these are the things that everyone can actually do today, not in 5 years or 10 years. Yeah. And they should be the foundation of what we know work in human today.
Yeah, I began my talk last week in Aspen by asking for a poll. Maybe we'll do it here. And that was, if you could live to be 123 years old and have your perfect health until you're 123, and then die immediately in your sleep at 123, who here would sign up? Who here would want that? All right. Yeah, anyway. Yeah, exactly. It's not enough. Well done.
It all depends on what you get.
Yeah, exactly. I want more than near. That's funny. Yeah. So, but I think that's this thing, is that right now everybody wants to extend healthy lifespan. But if we want to get beyond what we currently perceive as these human limits, we're not going to get there by exercise and diet. So the question is, what do we see as possible? What do we see as our goal individually and species-wide? Nir, let's talk a little bit about metformin and TAME. Maybe first talk a little bit about metformin and the work that you're doing on it, and then afterwards we'll talk more about TAME and its implications.
Well, the thing on metformin is, there are 2 things I want to tell you on metformin. Metformin is the first drug of choice for type 2 diabetes. It's been for 60 years. It's generic. It's safe. It's cheap, first of all, 690 pills for $60. And it's very safe, 60 years or billions of years of use. Okay, so this is the drug. And this drug is really a tool for us. And why is it a tool for us? Because when you give metformin to nematodes, there's increasing number. This year, there are like 30 papers on metformin in nematodes. You give them to nematodes and other animals, they all live longer. So it looks like it's in the middle of the biology of aging. And what happens in variety of studies in humans, some are clinical studies. When I say clinical studies, they were controlled or placebo-controlled or something like that. They prevent diabetes in non-diabetic people. In diabetic people, they prevent cardiovascular disease. And in association studies, they are associated with less cancer, with less Alzheimer, and with less mortality. And so it seems that not only it's part of the biology of aging, but it seems we have a lot of preliminary data that when you look at different diseases, you get effects, and all the effects are kind of 25, 30% effects. So this is why we chose metformin to go to this TAME study.
So why do you feel metformin has these effects?
So it's kind of what I tried to say before. I think we know what metformin is targeting. But the confusing thing about metformin is that there's a fight between groups of what it actually is doing. And the reason is when you give metformin to an animal, to humans, or around cells, if it fixes aging, it fixes a lot of things. So I presented this SMILE study where we took 15 elderly and we did placebo-controlled crossover study. We gave some of the metformin first for 6 weeks, and then we washed it out and gave placebo, or the other way around. And when we look at the transcript of their muscle, we took biopsies of muscle and adipose tissue. And when we did that, we saw a whole host of things that were changing, but not only in metabolism, because you think diabetes, but also gene repair pathways and many other pathways. And the nodes were what we know is in the midst of aging, like MYC, mTOR, some, I don't want to just throw things, but they all made sense. So if you fix aging, and by the way, the same happens when you give rapamycin or when you give acarbose, you get a whole host, and it's similar host of now the environment is better. And that's what we find with metformin.
So the TAME study, the metformin study, is bigger than just about metformin. It's about, in many ways, opening up the regulatory framework for all types of treatments for aging. Can you talk a little bit about TAME and its implications?
Yeah, and I would say, again, 3 things. By the way, it's in the Israeli army, you have to, everything is out of, there are 3 things for everything. So if you don't have 2, you have to
just make up. The third one is the same
like
as
the
the first
blankets have 3 sides, the one and the second, and then the width of that. So 3 things. First of all, we're going to prove that one drug, okay, that targets aging, affects appearance of several age-related diseases at the same time, okay? This is the proof of concept. The second thing is that we are working with the FDA. So at the end of this study, there is an indication of what is aging, okay? So that now it's clear and leads to the third part that now pharmaceuticals will have an indication where they can, and biotechs, and a template where they could go and develop better drugs, combination of drugs, where we could really expand health span much, much, much better. So it's really about paving the road for all you guys and for the future to have indication that we get over this problem that we have.
I think that's really important. I'm referencing what Ned said in his remarks, that there's not an indication for camping or playing a new instrument or doing all the things that younger people do. And yet, if TAME or any of these other interventions can show that they can treat aging, and the pathway to doing that is to say, well, if you're treating aging, you're also treating these known diseases that are correlated with aging, then that opens up a whole new world. Before we talk about the big picture unified theory, there's some areas that nobody here is doing specific research on, but are just fun to talk about. And one of the most interesting that's now starting to show up in the popular media is parabiosis. I don't know, does anybody here watch the HBO show Silicon Valley? But they have a thing where there's the blood boy, where they get, careful, where the rich Silicon Valley billionaire has the young guy who comes and they hook him up and they connect them for blood transfusions. But anybody who wants to talk a little bit about parabiosis, which has been happening for more than 100 years, but where the recent research, I know Tom Rando is here somewhere, but where that is heading and the implications of that work. Judy? Yeah.
We're working a little bit on this. I will say there are two parts to So first of all, parabiosis will probably go away. We won't have to be joined at the hip. There'll be blood exchange,
Darn. Yeah.
transferesis apparatus. But it goes two ways. So if you take what's in the blood of a young animal and you give it to an old animal, there are certain things that the old animal does better. They improve muscle repair, et cetera, et cetera. But the opposite is also true. The old blood given to a young animal causes a decrement in function. And that's very much less understood and very much less studied. So in order for us to harness what we know from the parabiosis experiments, we have to not only understand what the young people or mice make that help the old, we have to also understand what happens during aging that causes the decrement in function. And both will be necessary.
A publicity for AFAR is coordinating Nathan Shock Center. Our Nathan Shock Center have a core that provides parabiotic animals to anyone who wants it for $1,500. Okay, so it's you do young, old, and then the young and old together, and you get a triplet like that. It really was supposed to help a discovery of things that moves in the blood from one to the other, but the reason, and it will go away in this sense because we'll maybe find, the reason it's attractive for scientists, and I'll give this example, we noticed that the intestines of the young gets old in parabiosis from the old. Now, we discovered several mechanisms, and in order to show this mechanism, we can do the transgenic animal in parabiosis to see if we prevent this effect. So the parabiosis is actually a good model actually to test if you can block the aging or something like that.
And help identify what those bad things are and what the good things are, so eventually we don't have to do the transfusions.
Then that's why we didn't invite anyone from PETA to this event.
I'll just there's a variety of efforts happening now to essentially partition blood, purify from young blood, looking for the molecular identity of factors that by themselves do some of these good effects. And they're called circulating youth factors. So this is going to be an area where candidates are going to wind up in the clinic in the next 3 years. Anyway, I think it's going to be a very powerful area, both on the KIF side, that's the circulating youth factor side, as well as antibodies against what Judy was talking about, the so-called circulating aging factors.
Yeah.
Your mother had this face of you didn't tell her that. I
did. I actually don't think I have mentioned it to her. Yeah.
Well, that's really nice. This is the principle of bringing your mom to events. I should have brought mine. This is great. Welcome.
It's my understanding, I'm not an expert in this area, but if you exercise an old mouse and you inject it in a non-exercised mouse, right, the blood, then you get the same effect.
Yes, you can.
Yes.
Right. So it means that with exercise you can retune your system.
Yeah.
And I think Tom Rando, I'm not sure if he's here, he's doing very interesting work on trying to understand it at the molecular level, what these
This is one of the tragedies, is that everybody knows that exercise is great for you. It has all of these positive effects that we're talking about. But people would rather kind of be sewn together with a young mouse than to exercise 45 minutes a day.
But I think one point that Tom was making at a recent meeting was that if you're young and you exercise, it's pointless for your health. But if you exercise when you're middle-aged, you get actually the benefits, because then it restores apparently some of the functions or the deficiencies that your tissues and organs have.
Yeah.
So,
and for the first time, someone else can exercise on your behalf.
That's very funny. So just quickly, we have all these pieces. Let's just talk just a little bit. I don't know how much we can add to that. We have all these little pieces on the periphery. How do we stitch these? Is there a process for stitching these pieces together with a unified theory and a unified approach? Maybe, Ned, why don't we start with you?
I don't think there's a trivial way to stitch the pieces together. Largely by virtue of the metaphor that's been used in the room, whether you use the elephant metaphor or the tree, because you don't know the extent to which each mechanism overlaps and influences the other. My belief is that in 2020 hindsight, I would say 40 to 50 years from now, all of this will make sense. But in the meantime, I suspect that the progress we're going to be making both scientifically and then ultimately in the making of pharmaceuticals is going to be through a relatively reductive approach in which systems that produce aging effects, we figure out ways to perturb them, and we then bring them to the clinic. Now that's my sort of depressing view of this, and I'll give the sort of happier view. I do believe that we will uncover a mechanism that turns out to be a single most important mechanism, what people call the sort of underlying ticking clock. No one knows what it is yet. Candidates include things like an epigenetic ticking system that ticks forever and that gets reset every time you make germline. And it's possible that insights there over the next decade or two might wind up stitching many of these phenomena that seem like they exist as separate islands are in fact a single continent. But it's not clear if it's going to work out that way.
That's such an interesting point. Sorry, when I talk about this, the thing that really shocks people when I say to take a step back from what you know and just imagine this: you have 2-month-old. You get home from a vacation. You have 2-month-old milk in your refrigerator. And then you say, well, I'm going to make yogurt or I'm going to make cheese. And you use that milk. And then you have, oh, my God, it's fresh yogurt or fresh cheese. And yet we think it's perfectly normal that a 30-year-old man mates with a 30-year-old woman and they have a 0-year-old child. So there's something that's inside of our body that is resetting the clock. And that's why the Yamanaka factors work. And I think that it makes a lot of sense that if we can figure that out, there are some clues that we can follow to figure out how that clock works. Eric?
I'd like to point out there's an alternate sort of model also, which is right now the field has been moving in the last 15 years trying to identify these so-called aging pathways. Think about the TOR pathway. We have rapamycin that targets it. Think about AMPK. We have metformin. Think about the sirtuins. We have resveratrol and others. One of the things that, where the field is moving is trying to see how are these pathways talking to each other. And so one way would be to actually test combination, rapamycin and metformin, trying to say, does that work better than each individually? And so I think there will be a lot of activity in this field, but there's also another aspect to this, which is the more we study these pathways, the more we realize they are incredibly interconnected. Yeah. And out of this came the idea that maybe what aging is, or what the end of life is a collapse in these interrelated networks. And so there might be, for example, a key element in these nodes. There's a whole theory of network biology, which really posits that our cells are incredibly complex network, which means there are nodes in there. Once you start tinkering with these nodes, you might actually make the whole system collapse. And I think, to me, this is really an exciting area of the whole field of biology that we have barely touched.
Yeah, really. So bringing this back in the last segment of our conversation before we open up the floor, 20 years from now, somebody walks into a doctor's office and they say, Doc, I'm getting older. Can you help me? What does the doctor say?
Drink red wine.
Well, I think the amount of red wine that you need to drink, if the resveratrol theory even works, we should all get started now just as a precaution.
Well, it depends on how old you come in the doctor's office, I think.
It's a 20-year-old
50.
50 is pretty good.
Yeah.
I think then you probably, there will be some practical things or maybe drug-related interventions that you can have. But I'm not sure if you are already aged and you have extreme tissue remodeling going on, whether you can undo all that remodeling and bring it back to a youthful state. I have serious doubts about that. But I think that part of the question with regards, if you're interested in intervention, then you would have to understand, which aspects of the aging process are reversible, which ones are not, and how you could intervene with them. So I think there's a lot of laboratories that are interested in that and the question, and those answers will come. So yeah, it's an interesting question.
Yeah. Nir?
I feel like I have to change the mood here a little bit because it sounded to me all of a sudden that we're like, we don't know anything about aging. There's a lot to go. If a 50-year-old in 20 years will ask to live longer, maybe we can get to 50. I mean, we're past 80 now.
Yeah.
And in fact, Jan, especially for you, we see your movies here where you take animals and double their lifespan, and you're sitting here and saying, I'm not sure that in 20 years there'll be anything. And I really want to say that yeah,
this is the excitement we've been trying to generate.
We don't know a lot about aging, but we also do know a lot about aging, and we have been successful in labs across the world to increase health span and lifespan quite significantly. And not only that, I would say that we have, in some way, easier task than people who are coming like from the cancer point of view, because evolutionary aging is similar in all animals. All animals have this hair problem and this skin and the bones and everything else. And some of the drugs like rapamycin, you give it, to all the animals, and all of them are basically living longer. So I think the trunk here is not so big to start and understanding aging more, but it's not that we haven't been successful. We've been successful, and we should start right now to increase human health span.
So the pay lines at the NIH should drop, or should they increase?
So what's the question? I'm sorry?
No, I'm just saying, I think Nir makes the argument that, we already know a lot, and I'm trying to make the argument that we need to invest
Yeah, well, I think
in aging.
both of those are true. Yeah. Those aren't contradictory things. So definitely it's true that we know a lot. Definitely it's true, as we've all said, that there's a ton that we don't know. Definitely it's true that we are, if in a relative perspective, investing relatively too much on the diseases of aging and relatively not enough on understanding and addressing the underlying causes of aging itself. I think on those things we can all agree, and I'll add another one. Definitely we agree that until the rest of the world reforms, we need AFAR more than ever to help spark this process and make sure that, as Ned was saying, all different kinds of research into these and other areas has the opportunity to see the light of day, to grow from a small seed into something bigger. But maybe Judy and Eric and Ned, further thoughts on this?
I want to ask Nir a question. Do you believe there is a limit to human longevity? That's a loaded question because we have a mutual friend.
Yeah, a limit to human longevity. And I'll tell you my answer, and my answer is yes, okay? But that doesn't mean that we cannot change this limit eventually, okay? But I think what's appropriate, and it's very important for us, is to take those demographers who says, we'll just continue to live forever, and make them notice that there's actually a roof. So we might, and if we want to live forever, we have to invest in science in order to improve our health span. I think that's the thing. Now, whether it's 115 years or 120 years, and why is it a biological or a physical process, I don't know, but between 80 years old, which is life expectancy, to 115, we have 35 years that we can improve health span in. And let's start with that, and then we decide if we want more, and what does it mean.
Well, I also want to point out, think about mice and humans. So 30-fold difference in lifespan. We have roughly 97% genetic identity. So this begs the question of what did evolution do to take these very similar, genetically similar organisms and cause a 30-fold increase in lifespan? We don't know the answer to that. And I think, again, this could be a gold mine if we understood that better, but we don't. We really have no idea.
We don't, but as I was saying in my remarks, as we get the human population of Earth eventually sequenced, as we sequenced more and more animals, as we use big data analytics to compare genotypes and phenotypes, and as we use gene editing to knock out certain genes to figure out what they do, we're never going to understand that completely, but we'll understand more and more in an exponential rate of increase in our knowledge. Just some final thoughts, if anyone has, before we open for the Q&A.
I'll give you the 20-year, my prediction.
Yeah.
So I think, if you look at people that go to dermatologists as a proxy, I think that population, all of them will be taking metformin, okay? I take 1.5 grams a day, and I go to a dermatologist, okay? And there's a variety of people here who are not diabetic in this room who take it. I'm not thinking about anyone in particular. I think there will be a series.
You didn't just rat out your mother, did you?
Oh no, she's not on it.
Oh, okay.
Okay.
She wants blood.
Okay. You have no idea. Okay. All right. Sorry. It's funny. Haha. Okay.
I want your blood.
No.
Okay.
In defense, my mother is exactly the same way. When I told her about my conversations with Nir, she went to her internist demanding metformin. He wouldn't give it to her, so she went and did some more research about what she needed to say to get on the third visit. He finally broke down.
Okay. So anyway, I think there'll be metformin. I believe there will be a series of senolytic therapies 20 years from now, including, in all likelihood, a systemic senolytic therapy. Now, that's going to be a bit of a trick. What we're doing at Unity initially is all local. We will have systemic eventually. And in principle, that should phenocopy features of Jan and Judy's mice. Yeah. That's a big deal. And that's 20 years. Okay, that's something we can all think about and plan for. I believe, in all likelihood, there will be some acute use of rapamycin. Matt Kaberlein demonstrated recently that these very short, high-dose pulses seems to copy the effects of chronic use, and that effect is sustained after cessation of drug. And so that's just the stuff we know about. I think there'll be a series of circulating youth factors that are studied in various clinical settings and are approved. So let's tick down the list. It's 20 years. You're going to have systemic senolysis. You're going to have rapamycin acute. You're going to have everybody metformins in your breakfast cereal. You're going to have circulating youth factors. That's a lot. Things will be pretty different. And just that list I gave you doesn't require fundamental biological breakthrough, which we will have. So the list I gave you is the boring list.
Can I add to the boring list?
Yeah.
And this is an area that we haven't talked about, which actually addresses the whole issue, is there a maximum lifespan? The rejuvenation technology is a part of aging research. I mean, a lot of what we've heard is about slowing down the aging process in all of us, but there's incredible progress being made in the lab in terms of what we call rejuvenation technology, which is the idea of repairing. And think about aging, I think when you asked the question, I probably should have answered better. Aging is a loss of repair and maintenance. So we think of ourselves as sort of immutable, but we are constantly being repaired and turned over. Your bones turn over every 9 months, your skin every 21 days. So except for some very few parts of our bodies, a lot of us is constantly repaired and fixed. And I think the technology is now existing in mice, for example, to start repairing. You can, for example, induce a heart attack in a mouse, take a fibroblast from the skin, transform it into a stem cell, differentiate the stem cell into a heart cells, and reimplant it into the heart and fix the damaged tissue. So when we think about what Jan was talking about, when someone comes in old with a heart attack and a stroke and so on, there is today in the laboratory, in simple model system in mice, the ability it's being developed to actually fix, which is going to be an enormous aspect. We're far from going into clinical trials, except maybe for the eye, for macular degeneration. But I can see this as being, in 20 years, a field in which there will be a growing amount of interest in terms of fixing.
Yeah, I agree. And it's such an exciting moment. And I think as we've all seen from this panel, this is just the beginning. And the pace is only going to increase.